Okay, so for my project, I wanted to do something that was related to my personal research, and I tried to reproduce some results, but it actually failed, and I lost a lot of time trying to optimize it. So I don't have a lot of results, so I'm gonna spend, I think, most time on the introduction actually. But I'm still gonna try to show you some kind of nice videos and also speak about what could have been done maybe or could be done further. So I'm a student in the Laboratory of Physics of Biological System. So I'm doing biophysics, and I'm working on yeast. And one of my research topic is the DNA damage checkpoint. So I'll speak a bit about what it is. I don't know if you know anything about the biology of it, but basically, if you have the cell cycle, which is represented here, you have different phases. G1, G2 are the growth phase. The S phase is where the DNA is synthesized, and the M phase, which is the mitosis. And so basically, to make sure that the cell goes through those cycles in a healthy way, there are what are called checkpoints. So it's molecular feedback processes that basically would stop the cell if there is an issue. And the one I'm interested in is the DNA damage checkpoint. So here you have just yeast cells that are cycling, and you can see the nucleus and the division. It's really simple. So what happens is that if the DNA is broken or is damaged, the cell will stop at the DNA damage checkpoint and will wait until it is repaired, or if it cannot repair it, actually what happens is that it overrides the checkpoint. And that's what I'm interested in, the override of the checkpoint. So here you have a movie. So the cells, they're also a bit yellow in this movie, but don't really pay attention about the yellow. It's just something else. So you see first they're cycling normally, then I induce the break, so their DNA is broken. And when their DNA is broken, you will see they stop cycling. They're not budding anymore. And you will see the nucleus starts being stuck between two cells, and sometimes they will override. So you will see the division. Here there was one. I think there's one soon here. It's coming. Or maybe not. Yeah, so you see the division here. Okay. So in the lab, so that was before I was there, they developed a theory to understand the strategy of like why do they override, like what is the strategy behind this override. And so basically what they did is that they created a model that can then predict the override time for cells depending on the number of DNA breaks. So basically, I'm just going to speak about this formula. So what it is just the idea is that for you add penalties for waiting. So if you wait basically too long, so if you wait, you have more chance to repair. And if you repair, you can propagate healthily. If you don't wait, but you override the checkpoint, then you have less chance of survival because you don't, your DNA is broken. And so the idea is that cells that would wait too long, maybe it's good for them because they will be able to repair the checkpoint, but then by the time they waited, the first cells that waited, they had the time to replicate many times, and so they lose this competition, right? And this is kind of encapsulated in this. So A is just the override strategy. This factor is just the waiting um, penalty, and then there's the survival probability and the repair probability. And so the idea I had initially was to start my simulation with cells that would have different override time, and I let them repair or not, or override. And then what I would expect is that basically for one double strand break, I would see that the ones that were overriding at around 8 hours, so actually it's 7.5, I think, 7.5, they would be the one that are dominating the field, basically. And so the way I wanted to simulate this was with some kind of CA-like simulations. And so for this, I needed two things, which is the repair probability and the survival probability. And I just took them from the paper. I won't go into the biology, but basically from this, I can get the repair probability. it's actually the derivative of this curve that I fitted, and sigma is the survival probability. And for many breaks, then you would have sigma to the number of breaks. And so maybe I can draw it. So the idea is that if I look at the end of my simulation, the one that I repair should be, so the amount of cells, so if that's, okay, now I'll go to the next slide so that you understand maybe better. So the way I computed, I coded it is that each cell has basically 3 features, but I mean, 3 are not really important. That's just the way I did it. I could have done only 2. Anyways, the really important thing is what I call the genetic background, which is corresponding to the override time, and then the number of double strand breaks. Double strand breaks is just a cut in the DNA. Okay. And so the way the simulation works is just that each time there is a certain probability that it gets repaired, and if it gets repaired, then it can propagate normally. And then when it arrives at the override time, then the cell overrides, and here we check there's a certain probability that the cell will survive. And so basically, what I would expect is that if I plot so the genetic background, let's say, which is corresponding to the override time, then for the cells that are able to repair, I would get something like this. So that's the amount of cells. So for late override, I have more cells that repair, so I end up with more. Okay. And for the cells that are overriding, I get actually the opposite, because the earlier they override, the more cells they have. So basically, I have 2 curves like this, and then I would expect some kind of compensation. And if I look at the overall cells in the end, I was expecting something like this, where this would be 8 hours, which is the observed override time. So that's what I did. I mean, that's not exactly what I did. The first thing I did was I had some propagation, and then I see it didn't work, so I tried without propagation. So cells are fixed. But anyways, when I was using the real data, so the real probabilities, I got nothing interesting. So I think there are 2 reasons for this, which are that those numbers are extremely small, right? It's 10 to the power of 4 and 10 to the power of 5. Mm-hmm. So I tried to simulate a lot, much more. Still, it was pretty much flat. And even though I was like simulating and simulating, there was never a real peak. So at this point, I gave up on on trying to reproduce reality, and I just changed the way I was simulating this, because I had like all the code pretty much done. So I thought I would just do something different, and so I did basically kind of the same. So I'm not using the real probabilities. Now I'm just like randomly just using some kind of uniform probability, and it's much simpler. And also in the first simulation, the cells, once they override, they're dying immediately or not. And now I let them propagate, except that they have chances to die during the propagation, while the cells without that were able to repair, they can leave. And so about the rules, I don't know what I've said on that. So now there's propagation also. So the way I decided to propagate is basically that the new cell comes around the first cell, and there should just be an empty place, and it's randomly picked. And for the cell cycle time, for the repaired DNA, they have a normal cell cycle, which is 90 minutes. And for the one with double strand breaks, they propagate slower. So maybe I can show you now what it looks like, one simulation looks like. So for one double strand break, so it's really short. I don't know if you can see really well. So that looks something like this. So the one in a shade of blue, it's the ones that were able to override, and the ones in pink, they have DNA breaks. So you can see the one with DNA breaks, they have some little holes in the colonies. That's just because they can die. And when I did this and I tried to, it's not, I never know which one it is. And I checked, so the same plot I was drawing here. So that's after 24 hours. And the really interesting one is this one where you see that there's some kind of a peak around 8. So that's the overall. That's only the ones override that are repairing, and that's only the one that's overriding. So you see the earlier they override, the more cells they have. And yeah, we have a peak, and we have done just like more simulations. I accumulated them together, and we see that this is something kind of common. So that's what I was actually aiming for, but I did it in. I kind of cheated. That's not reality, right? But then I did the same with 2 double strand breaks, except that this time I did 24 hours. Also, something that I should have, I think I've done really in a really bad way is that if you remember the first probability distribution for the repair probability, I had it only between 6 and 16 hours. So I kept the 6 and 16 hours, but I think it would have been interesting to go before 6 to see what happened, if they were really like going down or if there was like another peak or something like this. But I can do it pretty easily. I just haven't done it. For the 2 double strand breaks, something quite surprising is that if I kept the same parameters, so not changing the probability, I get something that is just like going down. And that's not what we observe at all in nature. Actually, with 2 double strand breaks, they rest for longer time, which makes sense because they have more to repair. So yeah, I don't know exactly why, but I've tried playing a bit with the parameters because I thought the issue was basically, if you look here, for the repaired cells, they are pretty much no cells that are able to repair. So I thought that was the issue. And if I increase the repair probability, then I get something that kind of makes more sense, but still we don't have like a nice curve. And for this one, I also increased to 20 because the override time is much longer. So maybe I can show you what the simulation looks like. So with 2 double strand breaks, you will see there are only few cells that were able to repair, so you don't see a lot of blue cells. And so if I increase the repair probability, they're just gonna look like the first movie pretty much. And so initially, the idea once, if I was able to reproduce the reality, my idea was then to play with other parameters that we cannot really play in the lab with. So for instance, I was thinking just like nutrients, for example. I mean, that's kind of funny because someone did a presentation this morning, Cecilia. It was kind of like this also. And that's something I wanted to do. I did it, but it was like pretty much not really interesting. So, so I just done it. But yeah, see, for example, the effect of nutrients or maybe cell-to-cell communication, I thought would have been interesting. And so, sorry, that's actually the next slide. So yeah, communication between cells and like consumption of nutrients. And then, I mean, I do not work with cancer cells, but the reason why we're interested in the DNA damage checkpoints is basically because cancer cells, they're able to override many of those checkpoints. They have plenty of issues, they should stop, but still they're able to override them, and that's what we're interested in. And so I thought maybe using some other features that we don't see in yeast cells could be interesting with this kind of model. Anyways, I had the time really quickly to do something slightly different, which is this conjugative transfer of gene. That's something that doesn't happen in yeast, but is common, maybe not so common actually, depends. But some bacteria, they're able to transfer their DNA to other bacteria, usually plasmids. And so I thought, so you might be wondering why are they not able to repair? And the reason for this is that, so my yeast cells at least, there are many ways of repairing. And one of the most common way and the way that's the most efficient is called homologous recombination. And basically it uses, so if you have 2 chromosomes and there's one which is broken, use the other one as a template. And so I was thinking, what happens if you have bacteria that has that can transfer their gene and they have like a DNA break? Maybe some other bacteria could give the DNA so that they can repair it, because that's more efficient than other kind of repair. The repair mechanism that you see in this slide is called non-homologous end joining, and that's basically you have two ends like this, and they find somewhere to stick it together, but then that means you lose genes or things like this, and it's a really inefficient way, and not happening so often, as you can see. So I did something like this, where now if my cells are in contact, they can use the other one to repair. So if the pink cells are in contact with the blue cell, they can become blue. And I mean, it's kind of funny, I thought, because you can see, I feel like there's some kind of bridges forming between blue colonies. And well, this one was pretty long. I did another one with like 2 double strand breaks. But 2 double strand breaks, you have less cells initially, so it's a bit less interesting. It just feels like you have 2 big blue colonies propagating. And actually, this morning I was bored in a course, so I started coding something else with this, because I realized that looked a bit like a prey and predator, and I started coding it, but I don't have it. But I mean, the idea was that if, let's say, the pink cells are like wolves, so they are the predators, they need to eat other cells. So like when they eat it, then they would transform, like, as I said, the rabbit into a wolf. And I started coding this, but well, the thing is that the wolves are taking over, so I need to kill them. So I was thinking like something with overpopulation, I can easily reduce their level. Anyways, so yeah, I think that's it.
Questions? Otherwise, I have a question. So one thing I didn't understand too much is, so you mentioned this override of the kind of pause because of a DNA break. So is there a downside to this override, like in your CA, for example? Because it seems it just ignores the damage and continues on, right?
So initially there was a real downside, because it was after the override, it was will they survive or not?
Okay.
I pick a random variable. If it's lower than this survival probability, then
I
I
see.
die.
Okay.
But when I was doing this, basically, I would have like 1 cells every, like, I don't know, 10 to the 5.
That doesn't die.
Yeah, exactly. It was kind of uninteresting, I guess.
So the problem was that kind of overriding was too good of a choice because you never die afterwards, or too bad of a choice?
No, repair was just much more frequent.
Okay.
And the thing is that once you repair, your propagation is exponential.
Yes.
So it just takes over.
But so once you repair and you reproduce, can kind of your children, can they also get damaged afterwards? Or?
I didn't do this.
Okay.
I was thinking I could do it like in a later version.
Yes.
Where they can get damaged. Because the reason for this is that initially I wanted to reproduce the experiments, and in my experiments, they all have DNA broken, at least, I mean, in a controlled way. They all have 1 break in their DNA. Or
at the beginning? And
I actually induce it. In the movie, you don't see it, but
it just happened once. So the children, they don't have problems afterwards.
Exactly. And if they repair, then I see them repairing and then propagating. And that's usually really annoying because I cannot analyze them manually.
I see. Okay. Okay.
But we could imagine something like this. Basically, I was thinking that would have been for a later implementation. A common way, maybe not nowadays anymore, that's kind of old school, but the way we were inducing damage initially was with radioactivity, and that was absolutely not controlled, and you would have like 1, 2, 3, you don't know. And we could imagine something like this where we put a radioactive source somewhere, and then basically cells around would get damaged, and then they like switch. So from blue to red.
Okay.
I see.
So my other question was, so in your plots you have like 6 hours and 24 hours. So this is kind of Yeah, I was wondering. So your CA does not run in 24 hours in the end, right? Or
No,
does
no,
it?
that's the
Okay.
time of this. I mean,
Yeah, it's a rescaled time.
it's a rescale time.
Okay,
I mean,
okay.
I simulated like as if it was minutes, but
Okay, okay. I see. And okay, so
but like compared to this risk, I mean, this 24 hours kind of makes sense with respect to the time that you have here.
Right.
I mean, that's
Okay.
the idea.
Okay. Yeah, that's what I was just
Basically,
wondering.
when those ones are overriding, it's 8 hours proportionally.
Right. Okay.
And when I pick probability, it depends on the delta t also.
Right.
I mean, I just did the constants.
Yeah. No. I was just
which was not the case initially, and that's what failed.
Okay. Other questions?
I have a question about the cells that give DNA to others so that they can repair.
Yeah.
So in your life, it's like unicellular bacteria.
To be honest, well, I'm not a biologist either. To be honest, I did study physics, and I'm doing biophysics now, molecular biology. So I know basically only about yeast. I know that exists, but I have no clue whether this is actually used for repairing DNA. And I don't know a lot of what biology of bacteria except E. coli that I use. So that I'm not really, I mean, I cannot really answer your
And for
question.
yeast, like the normal one, like if you don't have the antibiotics, like does it happen like each cell has DNA breakage at some point in the liver?
Yes.
Or is it super rare?
Okay, is it rare or not? That's a good question. That depends on how you define rare, because the thing is, so we have common procedure where we let them naturally break their DNA, and and the thing is that we just use, I don't know, like 10 to the 7 cells. So maybe there are gonna be only few of them, but that's gonna be enough for us because then they're gonna propagate. But naturally it happens, yes. But usually it's repaired really well because there is homologous recombination. So in my case, they cannot use homologous recombination because there are these phases, and I induce the break at this point, if you want. So it's in G1, and in G1 they have only one copy of chromosome, so they have no template to repair their break. But if the DNA happens to cut here or here, for example, or here, but here actually I don't know what happens, but here or here, then they can just easily repair it. But that's actually a good question, because when I was coding, I started wondering a bit more about my own system that I'm using every day, and I realized that I actually don't know what happened between this and this. Because it's replicating their DNA, but their DNA is broken, and there are plenty of things happening on the DNA when it's broken, so I have no clue actually what happens. So that made me realize that I'm a bad scientist and I should read more papers. Yeah, anyways. It is actually. So.
I have one last question, not directly related to the project, but what is the size of the data set of such videos that you have? Just wondering, because we have some idea of project where we could train some CAs to kind of mimic biology videos, but so these kind of videos are very interesting. But
like, what is like how heavy is it? It is?
I mean, how many such videos do you have? Do you have like on the order of 10,000, or the order of 100, on the order of 10?
So we may need to work on this kind of things. We a lot to have like this kind of videos, and we have all kind of range of videos. For example, here, this is like the beginning of the movie, and there's something more movie.
Okay.
So we have some like 3 hours videos where you see them like growing a lot.
Okay.
My video is actually 24 hours because they are rest and they don't grow and they grow in size. But we have I don't know what is the data set, but I feel like 100, more than 100 can be easily. Okay. I mean, in the whole lab at least.
I was wondering.
Because I haven't done like 100 of those movies. It takes me like 3 days
to
have a movie, so I do not get. I mean.
Okay. Well, I may contact you or your lab at some point to get some of these videos.
I mean, for sure. I mean, we've been giving them a lot, so.
Okay, great. Okay, thank you.